This is a walkthrough of our small Genome browser. This is the interface you get after you've selected, loaded up, and aligned data. On the left-hand side of the screen, you can see the genome sequence view. So in this case, we're looking at an H1N1 genome. The first accession number is the variant that we're actually seeing the codons for. The second accession number is the variant we did a comparison with. So we can see changes between the two of them. So H1N1, you can see, has segments. It's a segmented single-strand, negative-strand RNA virus. In this view, and we can just zoom in by lassoing any area on the screen, codons are represented by thin rectangles of color. So here's the start of segment 2. You can see a short, untranslated region. Here's the start codon for the PB1 gene. And then here's the start codon for the PB1F2 overlay gene. You can see this is a frame-shifted overlay. And here you can see better the thin rectangles per codon. So you can see, here's the start codon, and you get a little tooltip flyover for each codon. So it shows you what segment you're in, the name of the gene, the product, the codon itself, the nucleotide offset from the start of the segment. Likelihood is an R calculation, we'll see in a minute. GC% we calculate per gene. KAKS we calculate based on the comparison with the other variant, as well as the Kimura two-parameter evolutionary distance metric, the K80 metric. So these thin rectangles pile up to form genes, and then the genes are concatenated to form segments. So one thing you might want to look at if you've done a comparison is where the changes were. So you can do a search in all the text for each codon to find synonymous changes, and then we'll do a search for non-synonymous changes, and we'll highlight those in blue. So now I can see the distribution of changes across the segments. So we could see segment 1 had only synonymous changes, right? So his KAKS is actually 0. Also, segment 7 and segment 8 had only synonymous changes. So one thing that might simplify what we're doing is we can get rid of everything that isn't highlighted. So here's all the codons which we had highlighted, and we've gotten rid of all the rest of them. So we've just zoomed into strictly the changed codons, strictly the highlighted codons. So while we're here, we might be interested in analyzing, say, the transversions versus transitions. So now I've done a search and I've highlighted transversions. So I can see from this display the location of all the transversions, as well as I get a visual idea of the ratio. But then I can look at the bottom, this line at the bottom, where I can see quantitatively there were 45 changes altogether, and 8 of those were transversions. So there's about a 1 to 5 ratio of transversions to transitions. So that's an example of doing a search. One thing you might be interested as well is we can color by other things. Here it's colored by gene product. We can also color by GC%. So using a linear color map, we don't get much difference between, so here I can see the GC% is 41, here it's 44, here it's 44. So using a linear color map and just looking at the GC%, I don't really see, significant differences. But we can be more quantitative and probabilistic about this. This is an R interface. You can actually type in any valid R statement using any of these variables which we surface from the underlying data. Or we could choose this one which is already primed. So here I want to calculate the probability of seeing on a per gene basis this GC count given this nuke count per gene, if the probability of seeing a GC was the overall probability. So I want to see what exactly this probability is on a per gene basis. So now we're coloring by likelihood, and you can see this looks a bit different. So if we look at segment 5, his GC percent was 46. The overall is 43. So the likelihood of seeing that was 1.5%. So that's pretty much on the borderline of where we would reject that this is just a random fluctuation. But if we look at gene M1, his GC percent is 48, which is a lot higher than 43. And so the likelihood of seeing that GC percent given his number of nucleotides is actually 0.002. So that's pretty much beyond what we would accept as just a random fluctuation. So this was a segmented virus. Viruses have pretty diverse structures. We can look at a couple other examples. So here's a hepatitis B virus. So hepatitis B is a circular, partially double-stranded DNA virus. So one interesting thing, so since it's circular, the top codon and the bottom codon in the display are actually physically contiguous. So you can see this large area of sort of tannish purple and this large area here are both part of the P gene, which codes for the polymerase. So you can see the P gene goes almost 80% of the way around the whole genome. Every other gene actually, at least partially, overlaps the P. Here's the X. Here's the core protein, here's the X gene, and then here's the surface protein, which is totally overlaid on the polymerase. And if we zoom in, we can see that it's actually an out-of-frame overlay. Here's the AT and this histidine, G and this glycine form the start codon for S. So we expect that S has quite a bit of evolutionary pressure on it, since that's the surface protein. That's the thing that the immune system uses to target the virus. And the polymerase also has environmental evolutionary pressure because that's the target of antivirals. So we could look at the number of changes in this area to see if they manage to change in this frame shifted overlay or not. And it seems like they do change, but if you look at it visually, it seems like there are fewer changes here than there are in the rest of the genome. So that's a visual intuition. We can be more probabilistic and quantitative about it. So we could select this area where P and S overlap, and now I can do a calculation based on just selected screen data. And what I want to see is the likelihood of that few changes or fewer, given that changes were, the probability of seeing a change was the overall probability of seeing a change in the genome. So let's just calculate that. So now we're coloring by likelihood in the selected area and by gene everywhere else. So the likelihood of seeing this fewer changes in this area is 6.5 times 10 to the minus 7. So that's beyond the probability of that we would accept for random fluctuations. So, but it is managing to change. So we can zoom in and just see, well, how do they manage to, how do these codons manage to change when they're shifted like this? So let's just zoom into this area. And now we can see, so here's a case where it's a position 3 synonymous change in the S gene, and it's a functional non-synonymous change in P. Position 1 is changed. Here's another synonymous in S, non-synonymous in P. Here's another synonymous in S, non-synonymous in P, and then a double synonymous. So here's a region where P is changing and S is just seeing synonymous changes. And now if we scroll down a bit, we can see here's the opposite, where here's a synonymous change in P, non-synonymous change in S, so it's position 3 in P, position 2 in S. Here's another one, position 3 in P change, synonymously, functional change, position 2 in S. Here's a double synonymous change. So even though these two genes are overlaid and frame shifted, they still manage to evolve by one making a synonymous change where it's non-synonymous for the other. So we can look at one more virus, I think. So here's SV40. One interesting thing about SV40 is it's got both sense strands. So it's a double-stranded, it's also circular, so the top and bottom codons are contiguous. And we can see that we've zoomed into the GP5 gene, so here's the start codon for the GP5 gene. And if we just scroll down, we can see here's the stop codon for the 3 and 4 protein genes. So this is the traditional start at the top, stop at the bottom. But if we scroll down a little, so here's GP5, we can see, so here's the end of GP5, traditional stop codon, some non-coding, and now here's GP6, so here's his stop codon, so we had to reverse complement this sequence. So this is going the other way. Here's the stop codon, and if we go to the bottom of the GP6 gene, we'll see his so actually now we're in intron region, so here's the stop for another backward sense gene, and if we go to the bottom, now they're overlaid, and now we can see here's the start for GP6, and here's the start for the other. So it has genes that go positive and negative in this circular genome. One other virus we can look at really quickly is Schmallenberg. So Schmallenberg, also segmented RNA negative strand virus, has L, M, and S, obviously, for large, medium, and small. Now the M gene produces this polyprotein, which once it's in the host cell, it's cleaved into GN, NSM, and GC. So this is just some more structure that we can visualize, because you might be interested in investigating there might be differences in the structure based on what's going to happen in once it gets cleaved. And then you can see the S gene here has a start codon, and then it has an overlaid alternative site for initiation. So it could be there's some leaky scanning going on here. So that's the left side of the screen. On the right side, we can see the codon statistics view. So codon statistics gives you the distribution of codons in the overall genome. So here you can see that there's a lot of lysine in this organism. And if I open this, I can see the distribution of lysine per gene and per segment. So this codon statistics gives you overall codon distribution, then distribution of that codon within genes. I can look at the gene statistics view, and that gives you a gene view. So I see the distribution of codons in genes. So segments are sorted by codons. And then if I open the segment, I can see each gene. So it's sorted by genes if there's more than one in the segment. And then if I open each gene, I see the distribution of codons in each gene. So it's a more quantitative way to investigate the composition of each gene as far as codons.